Exchange of oxygen and carbon dioxide in organism takes place in two locations. During international respiration, also cellular or tissue respiration, the gaseous exchange occurs between blood and tissues or cells of the body. During external respiration, the gaseous exchange takes place between the blood and the external environment. Blood serves as a transportation medium for carrying oxygen and carbon dioxide. The body structures which are needed for gaseous exchange between the blood and the surrounding medium are known as respiratory organ. Depending on the type of medium, we have two principal types of respiratory organs: gills for aquatic respiration and lungs for terrestrial respiration. The same animal may have both gills and as well as lungs present in accessory respiratory organs, are also same vertebrates, are also same invertebrates. So in lungfish, in larval amphibians, we can see both gills as well as lungs. All respiratory structures consist of a moist, semi-permeable and highly vascularized membrane exposed to the external medium, so the exchange of gases takes place by diffusion. Gills or branchia are the aquatic respiratory organs of fishes and amphibians. Embryos do not utilize gills at any time in the embryonic or adult life. In addition to gas exchange, gills may serve for loss or gain of water and elimination of salts in marine tellures. On the basis of the location, gills are of two general types: internal gills and external gills. In some, both internal are present. Internal or true gills. Internal true gills are characteristic of fishes. They are located in the gill slits and attached to the visceral arches. In embryos, embryonic pharyngeal pouches don't open by gills outside in there, so that no gills are present in them. Gill slits. Gill slits are one of the most fundamental layers of the chordata in the embryo. The pharyngeal cavity is connected to the outside by a series of lateral openings known as the gill slits. So you can see the gill slits here. Sorry, those are not the gill slits. These persist in the adult state of protochordate cyclostomes, fishes, and certain amphibians, but become reduced, abolished, or modified in higher vertebrates like for us. Number of gill slits varies in different chordates: 140 in Amphioxus, 6 to 14 pairs in cyclostomes, 5 pairs in most elasmobranchs, 4 pairs in chimeras, 5 pairs in most bonificia, and 4 pairs in some teleos. The gill slits are separated from one another by partitions called visceral or gill arches. The structures are supported by skeletal structure of splanktocranium together forming the visceral skeleton. Structure of true gill. True gills are developed on the walls of gill arches. You can see the gill arches here. So true gills are developed on the walls of gill arches. Typically, a gill is composed of two rows of numerous gill filaments or lamellae. They derived from epithelium on either side of an interbranchial septum containing arteries and supported by the branchial cartilage or bone of a gill arch. Single row of lamellae on one side of branchial septum forms only half of the gill, called a demibranch or hemibranch. A septum with two attached demibranchs comprises a complete gill or hollow branch. Gill filaments are richly supplied with blood capillaries, and it is here that exchange of gases with water takes place. So in gill filaments, the exchange of gases with water takes place. Operculum, it is in a bonifish, a bony flap called operculum or gill cover, arises from the hyoid arch, opens by a single slit like crescentic external gill opening from behind, and covers the gills in a common opercular cavity. So interbranchial septum, the medium septum is best developed in elasmobranchs, reduced in some intermediate fishes like chimeras, greatly reduced or perception in teleos. You can see it here, the interbranchial septum. Spiracles in elasmobranchs and ganoids, the first gill slit between mandibular and hyoid arches bears a reduced pseudobranch and opens to outside through a small opening, the spiracle. In chimeras, lungfish spiracles become either closed or lost in the adult. Reduction number of embryos. Number of gills greatly varies among fishes. 7 pairs in Heptanchus, 6 pairs in Hexanchus, and 5 pairs in most branchs in addition to spiracle. External larval gills. As against true gills, the external gills are formed as branching outgrowths from the exposed outer epithelium or gill arches and not from that of the pharyngeal pouches. They are ectodermal in origin and usually temporary organs found only in larval stages, also termed larval gills. They occur in the larva of lampreys, fury bonefish including polypterus, lungfishes, all including Sicilians. In amphibians, larval external gills are absorbed at the time of metamorphosis, but in water-living mad puppies as well as larvae, both external gills and gill slits are passed during adult life. In some aquatic salamanders, gills are absorbed but gill slits remain. Then we go to read thoroughly vascularized biotic arches. External gills are simply webbed in water and no spit water can pass through gill slits in case of true gills. As you can see some example in the lungfish, you can see the external gill. You can see axolotl larva external gills, in tadpole of frog external gills, salamander.